Good morning, everybody, and thank you so much, Professor Glazerman, for this very kind introduction. And I think we are thrilled to be on the third day of our wonderful meeting, of speakers-only meeting here on epigenetics here in Florence. And I want to thank again Giuseppe Menarini Foundation and Marianne Legato for putting us all together and having the possibility to be here and to lecture, learn, and teach. The topic of my lecture is epigenetic aspects of reproduction and early pregnancy. Moshe Schiff, that was with us 2 days ago, during the last 2 days, is not with us today because he is a religious person and he's on Shabbat on Saturday, he's not going by cars, but his spirit is with us. And I want to show you a very short video from YouTube that he gives us the perception and the understanding in a TEDx lecture that he had in Bratislava, what does epigenetic means.
So it all came to life in a dark bar in Madrid. And as I was stepping into the bar, I encountered my colleague from McGill, Michael Meany. And we were drinking a few beers, and like scientists do, he told me about his work. And he told me that he is interested in how mother rats lick their pups after they were born. And I was sitting there and saying, this is where my tax dollars are wasted, on this kind of soft science. But as the beer got more intense and the alcohol gets into the brain, you become more receptive. And he started telling me that when the rats, like humans, lick their pups in very different ways. Some mothers do a lot of that. Some mothers do very little, and most are in between. But what's interesting about it is that when he follows these pups when they become adults, like years in human life, long after their mother died, they are completely different animals. The animals that were licked and groomed heavily, the high licking and grooming, are not stressed. They have different sexual behavior. They have different way of living than those that were not treated as intensively by her mother. So then I was thinking to myself, this magic? How does this work? I'm a biochemist. I believe that there are chemical explanations to nature.
So, this is the presentation of Moshe, what does epigenetic means. And I divided my lecture in 2 parts, molecular and basic epigenetics, and afterwards clinical epigenetics. If we are looking at the definition of epigenetics from Wikipedia, it's a study of heritable phenotype changes that do not involve alterations in DNA sequence, changes that affect gene activity and expression. Such effects on cellular and physiological phenotypic traits may result from external or environmental factors or be a part of normal development. So changes that are done but are not involved nucleotide sequences. And the changes are, as we know, DNA methylation and histone modification. Gene expression can be controlled through the action of repressor proteins attached to silencer regions of DNA. And the changes can be through cell division for duration of cell's life and can be for multiple generations. These are the mechanisms that all of us know. I want to sharpen them. Epigenetic mechanisms that cause these changes, development in uterus, reprogramming that we discussed so much during the last 2 days, especially after Professor Glazerman's lecture, environmental chemicals, drugs, pharmaceuticals, aging, and diet. And this is the histones with the histone modification and DNA methylation. And health endpoints are cancer, autoimmune disease, mental disorders, and diabetes. The mechanisms are a very long list. Covalent modifications, RNA transcripts, microRNAs, messenger RNA, small RNA molecules, prions, which are proteins produced during infective processes in our body, nucleosome positioning, the steric positioning, and the genomic architecture. And the implications can be, and shown in twins that we are going to see later, genomic imprinting, cancer, diabetes, and influenced by parental nutrition, early life stress, addiction, and depression. Let's look on one of the mechanisms, microRNA. Acts after transcription by destabilizing transcripts or by inhibiting protein translation. MicroRNA control embryogenesis, cellular differentiation, proliferation, cytokine production, and apoptosis. And you can see the action of histone and DNA methylation, transcript, microRNA, and transcript destabilized. Altered levels have been associated with cancer, cardiovascular disease, viral infections, neurodegenerative disease. Let's look at early pregnancies. And let's look on the pathological pregnancies. Hydatidiform. Hydatidiform mole, it's very interesting presentation, clinical presentation, because sometimes we have a full molar pregnancy, and sometimes we have a partial molar pregnancy. So what's happening? The methylation of many genes occurs in primordial germ cells. Histone replaces protamine. After fertilization, paternal genome is demethylized. Some genes are imprinted for trophoblast formation. If we're looking on the oocyte, the ovum, in terms of epigenetic reprogramming, the methylation completes by development of the morula, and remethylation occurs when cells differentiate to inner cell mass and trophoblast. So here is the explanation. Why sometimes it's partial mole and sometimes it's full molar pregnancy. If male pronucleus dominates, androgenate is formed. This is the androgenate. Imprinted genes from sperm cause placental development. Degenerate to complete hydatidiform mole with trophoblast elements, but not embryo. So if the imprinting is done mainly by the male pronucleus, no embryonic tissue in this molar pregnancy. This is the full mole. But if male pronucleus dominates, created by excising male pronucleus, a genotype is produced in which embryo elements form but not placenta. So this is the mixture, and this is the explanation for the partial mole. Let's look on pathological pregnancies. Genetic aberrations, trisomy 16. Trisomy 16 is always incompatible with life. In this case, cytotrophoblast is hypermethylated. Extra chromosome influenced epigenetic profile of the genome changing to the level of methylation of cytosine per guanine domains, sites of all chromosomes. However, biggest number of differentially methylated DNA found on chromosome 16. Let's look on trisomy 21. These survive. We have Mongoloid children born. Why part of them are aborted and why part of them are born? Epigenetics. Why are most trisomy 21 embryos aborted and some born as Down syndrome? Genome-wide methylation studies have shown epigenetic signatures of Down syndrome in skin, fibroblasts, buccal smears, livers, and placenta. Chromosome 21 contains genes for DNA methyltransferase 3, which is very important, and he is a key player in epigenetic regulation. So everything is dependent on epigenetics. If this embryo of trisomy 21 is going to be aborted, or a trisomy 21 baby will be born. Epigenetic dysregulation due to dose imbalance of chromosome 21 during brain development may influence cognitive impairment. It's not only to live or to be aborted. It's the degree of the influence on cognitive development. And we know that these 21 trisomy children, they have problems in terms of cognitive development. Does excess DNA on chromosome 21 affect cognitive impairment on other chromosomes, as chromosome 16? We still do not know. Let's look vice versa. Monosomy, Turner syndrome, 45X. Only mosaics survive. Again, only mosaics survive because the full Turner syndrome, they are aborted. We don't see them, or very rarely. If all cells, 45X, miscarriage. One of the studies that investigated 33 Turner syndrome patients found globally changed methylation patterns of all chromosomes. Hypomethylation occurs chromosomes and areas of hypomethylation in Turner syndrome, and preferential enrichment of chromosome 1, 11, 17, and 22, preferential hypomethylation of proximal promoters. And this preferential methylation and epigenetic changes show the extension of the syndrome. It's a syndrome. It's not only streak ovaries and webbed neck and short stature. It's coarctation of aorta, bicuspid insufficiency, and the whole series and presentations of Turner syndrome. APLA syndrome and development of the trophoblast. Antiphospholipid antibodies. Yesterday, our Italian colleague mentioned that there are changes, epigenetic changes in APLA syndrome, bless you, and presented that there are changes in MTHFR and homocysteine that can cause APLA syndrome. Antiphospholipid antibodies inhibit trophoblast differentiation in vitro. This may affect invasion, implantation, placentation, significantly reduces HCG release, early embryonic development. And it's associated with reduced leaf. We know, all of us know very well that leukemia inhibitory factor is essential for implantation. So, DNA methylation profile of antiphospholipid syndrome, not yet characterized, but 17 hypomethylated and 25 hypermethylated CpG sites identified compared to healthy patients. Lopez and Pedrera showed that genes associated with human pregnancy that are hypomethylated in APLA, in antiphospholipid syndrome. And here it is. This is the microRNA activity that causes these epigenetic changes and causes the whole presentation of APLA syndrome, which changes in the blood vessels, the impact on the trophoblast, starting from the blastocyst, and the very severe cases, some of them, of antiphospholipid syndrome. You know that after treating IVF and replacing embryos, we give huge amounts of progesterone. And progesterone reacts with membrane receptor releasing signaling cascades. Let's see if progesterone in such heavy doses can be a protective factor against epigenetic changes. So the connection between progesterone and its receptors causing intracellular signaling cascade and changes in nucleus in transcription. Receptor links to transcription factors and the hormone response element on promoter to acetylate histone and start transcription. Transcription factor proteins that control transcription from DNA to messenger RNA by binding to specific DNA sequence. So maybe it's some protective aspect by giving progesterone, and maybe we can save part of the epigenetic changes, and maybe it's sort of therapy to prevent epigenetic changes after assisted reproduction, and we're going to discuss later. Now, LH. You know that after, there is a protocol that after embryo transfer, except of progesterone, we give boosters of LH, Ovitrel, Pregnil, or whatsoever, to stimulate the corporal lutea to produce progesterone and to improve the implantation and to increase the implantation potential and to bring more and more pregnancies and deliveries. So at ovulation, LH induces expression of progesterone receptor, and VGA only secreted by the decidua, which expresses progesterone receptor. However, possible epigenetic effect of progesterone supplementation unknown as yet. HCG. Can we do actually something with HCG to prevent epigenetic changes? In repeated implantation failure, intrauterine HCG infusion increases endometrial markers of receptivity. HCG treatment of endometrial stromal cells, they actually promote the markers of implantation, and the methylation is reduced. So might be giving HCG and giving progesterone, it's a matter of longitudinal studies on many IVF pregnancies and various indications, various ages. You know, it's a variety of possibilities. If we're looking on sporadic or transgenerational epigenetics, Lacalle and Ventura defined already many years ago 3 types of epigenetics that we discussed yesterday, the day before yesterday. So this is a direct epigenetic, direct experiences from the environment, and the influence of the environment, intrauterine changes that Mark explained us so beautifully yesterday, and across generations. Defined changes inherited from parents, grandparents due to events occurring long before conception. So direct epigenetics, we know all the factors. I'm going to go into it when we're going to speak on the clinical part. And fetal factors consist of the hazards that cause intrauterine restriction, such as fetal hypoxia, asphyxia, placental insufficiency, prematurity, low birth weight, and drugs that are given to the mother. Fetal programming, very shortly, because we discussed it very profoundly yesterday. Environmental factors, diet, pollution, smoking. And this is the aspect that we so are interested, and we discussed it yesterday several times, on psychopathologies, anxiety and depression. Anxious individuals have higher global methylation levels than non-anxious controls, and this is influence the fetus in terms of changes in his life in this aspect of mental development and state. Adverse early experiences can affect glucocorticosteroid receptors. We're going to go into it later. Gene NR3C1 expression predisposing to anxiety and depression of the newborn. So the question is, does treatment affect epigenetic mechanisms? HCG, anticoagulants, progesterone, maybe immunoglobulins, when are they given in IVIG during the Apla syndrome? It requires investigation, but we should think about it and do something, because the epigenetic changes are tremendous. Again, a very short video that summarizes everything that was told till now. I love the
Here's a conundrum.
lectures.
Identical twins originate from the same DNA, so how can they turn out so different, even in traits that have a significant genetic component? For instance, why might one twin get heart disease at 55 while her sister runs marathons in perfect health? Nature versus nurture has a lot to do with it, but a deeper related answer can be found within something called epigenetics. That's the study of how DNA interacts with a multitude of smaller molecules.
So, to take home messages from this part. Epigenetic mechanisms are responsible for embryogenesis. Gametes and embryo demethylate and methylate anew. Some epigenetic signatures escape demethylation. Dosage compensation may determine compatibility with life in cases of embryonic aneuploidy. Dosage compensation may explain miscarriage of trisomies 16, 21, 45, hydatidiformoles, etc. And Apla syndrome may be associated with epigenetic dysregulation due to microRNAs. And medications like IVIG, progesterone, anticoagulants, and HCG required investigation into their epigenetic factors. What about clinical part of epigenetics? Allegra Torres, 2011, lifestyle. Everybody is speaking about lifestyle in any aspects of our health, but especially epigenetics. And he is mentioning the nutritional factors, physical activity, tobacco smoking, pollutants, alcohol, emotional, shift works. We as physicians, our shift works are killing us. They are doing a lot of epigenetic damage, because when you don't sleep, when you don't eat, and especially when a pregnant resident is running 3 o'clock in the morning in OR to do a cesarean section or to treat ectopic pregnancy that ruptured, it causes tremendous changes to her embryo. So he's actually summarizing the environmental toxicants, the individual characteristics, the lifestyle, and the settings. So epigenetic marks and lifestyle factors, including nutrition, behavioral stress, physical activity, working habits, smoking, and alcohol consumption. And again, transgenerational epigenetics. It can help explain how gene expression is modulated by lifestyle and environmental factors that actually show of the individual response to environment cues and acquired risk factors. And the lifestyle is part of our life and our epigenetic changes. Increasing evidence show that environmental and lifestyle factors may influence epigenetic mechanisms such as DNA methylation, histone acylation, and microRNA expression. Biancamoto 2017, again, it's the disease origin, developmental origin of health of disease that Marek and others discussed this very profoundly yesterday. And epigenetic mechanisms. There are potential mechanisms behind development programming in the context of DOHA. Events early in life during the development of particular organ can program that individual for increased risk of chronic disease later in life. And we'll come afterwards to the work of Boris that showed that many markers, epigenetic markers that were found in newborns were actually dissolved and disappeared in elderly life. The question is, if they disappear in the ages 22 to 35, if they can be presented later on still in diabetes, hypertension, and cardiac disease. So epigenetic changes in humans, it's very difficult to study. However, animal studies provide the opportunity to study all these mechanisms. And the role of epigenetics in developmental programming has been greatly advanced due to animal studies. And it shows how these changes can show disease later in life. Let's talk about epigenetics and ART. We discussed part of the things in my previous lecture on congenital malformations and epigenetic and genetic changes in newborns after ART. Fantastic study that shows us how the handling of the gametes and how the embryonic creation in the laboratory system can have epigenetic changes that will dictate from this early embryo if this human being in future is going to be healthy or sick. Adjuk, she is a fantastic scientist from Warsaw, from Poland. And she's collaborating with her colleagues in Cambridge. And you know that one of the biggest possibilities to study the whole developmental process of egg-sperm interaction, embryogenesis, and development of the embryo is embryoscope. Embryoscope is an incubator that contains camera and documents the fertilization process and embryonic development for 5 days until the blastocyst. And today, with artificial intelligence, we can pick up the best embryo by this mode of technology that we transfer in order to get the higher implantation potential and increase the pregnancy rates. So they are doing it on mice. And they discovered that during the fertilization process, the cytoplasm is not inert of the egg. It moves. And they presented the movement of the cytoplasm during the fertilization process. And they checked it when the sperm was injected under the zona pellucida, or straightforward by ICSI procedure that was invented by Palermo, that we heard him 2 days ago, from Cornell, from New York, or some other activators. And they show that fertilization-induced cytoplasmatic flow are a conserved feature of eggs in many species. The importance of the cytoplasmatic flow has been unknown, but advanced particle image velocimetry, fertilization induces rhythmical cytoplasmatic movements that coincide with pulsations of the protrusion forming above sperm head. We find that these movements are caused by contractions of actomycin cytoskeleton, triggered by calcium oscillations induced by fertilization. And her next study, 2 years later, shows fantastic thing that give us clue how we can choose during the fertilization and the movement of the cytoplasm the best embryo for embryo transfer. And again, this is my studies, but it's today is starting to be applied to humans. Again, documenting fertilization and the embryogenesis every 15 minutes by embryoscope. They documented the spikes of the cytoplasmatic movement, and they showed that there are two sorts of movements. Chaotic movement of the cytoplasm, and the vector is linear. And look what's happening. And this was proved because they are doing IVF on mice. When the fertilization cone, the point where the sperm conjoins with the oolemma, is protruding, and the vector of the cytoplasmatic movement is straightforward, this is the ideal embryo. But when the cone is flat and the cytoplasm is chaotic in terms of movement, this is the better embryo. And they showed that only these embryos in mice implanted. So, can you imagine how epigenetics influences all these procedures? The culture media, the injection process, if it's ICSI, the fertilization process, the sperm penetration connection to the receptor on the zona pellucida and to the oolemma. It's incredible. And everything surrounding this process is epigenetics. So combination of genetic testing of the polar body and examination cytoplasmatic flow is especially promising, and embryo quality within several hours after fertilization can give us clue what is the best embryo. And we know another thing, that we discussed it and we're going to discuss it again. We know that with long embryo culture, we have more epigenetic changes and we have more effects on the health and development of the embryo. Do you remember I told you about blastocyst transfer versus cleavage stage transfer day 3? And a shorter period between in vitro fertilization and embryo transfer may be beneficial as prolonged in vitro culture of embryos alter their epigenetic modification and gene expression. So, again, a big dilemma. Many laboratories, and most of the laboratories, they transfer day 3 cleavage stage embryos and they transfer blastocyst transfer, and the book is still not written. We do not know finally what is better in terms of epigenetic impact on this specific question. Pingborg, 2011, Epigenetic and Assisted Reproductive Technologies. Again, he's looking in the 4 syndromes, imprinted gene defects, Bacon-Wiedemann, Silver-Russell, Engelmann syndrome, and Prader-Willi. And again, he's concluding the children conceived by RT have altered lipid profile. We discussed it yesterday, fasting glucose and body fat distribution and cardiovascular function. So singleton conceived after fresh embryo transfer, actually, they are different that singletons are conceived after frozen thawed embryo transfer. And it has been shown that fresh embryo transfer, it's medium weight of the neonate. The frozen embryo transfer, the weight is much higher, up to 4.5 kilos of the newborn. This is epigenetics. This is epigenetic changes during the cryopreservation. And today, it's a big question if the vitrification of the oocytes, sperm, and the embryos, what epigenetic changes did they cause? We have clinical presentation. And another thing, if the woman has actually conceived first time from fresh embryo, what happens on the first attempt of the IVF, she delivered a child, and afterwards she went for the second cycle. She wants another child from frozen thawed embryo. The weight of their second child is going to be significantly higher because it was cryopreserved. So, the embryo implantation performed during freezing and thawing may cause changes in the development of the fetoplacental unit through epigenetic modification. Huntress, 2018, Epigenetics and Reproductive Medicine. And again, he is mentioning the animal studies, including embryo culture, superovulation, maturation oocytes, micromanipulation, and so on. And this is again the big enigma. What does cause these epigenetic changes? It's the technology, or these are the patients? This is their infertility caused sickness, because those people are sick in terms of infertility, because they cannot reproduce otherwise. And at least at this specific moment, you know, the women with unexplained infertility for years, if we are not going to treat them, suddenly they get pregnant. They are going to a trip on a boat and coming back pregnant. So we have the hypothalamic factors. But in general, it's sort of a epigenetic impact all the time. But it's question if these are the patients with their underlying disease or the manipulation or both. So superovulation can increase these changes. Whether epigenetic disturbance is caused by ART or an epigenetic error in the gametes is unclear. But it is possible that in some cases, assisted reproduction exacerbates pre-existing defects of gametes. Maybe the gametes, they have pre-existing defect. And by doing in vitro, throwing the sperm straight to the cytoplasm, handling the oocytes, culture media, we provoke these changes. So there is evidence that epigenetic differences and gene expression changes in ART cohorts with compared with those naturally conceived through genome-wide studies are required to confirm. Barberet. What impact does oocyte vitrification, this is what I told you before, have an epigenetics and gene expression? You know that today, many women cryopreserve their oocytes, what we call social freezing. We drop the terminology of social freezing. It's ethically, it's not nice. It's sort of freezing for future use. And the big question is if the vitrification process on the oocytes, the cause actually changes. So this study investigated DNA methylation, microRNAs, and histone modification in humans and in animal studies. And in humans, when they checked the DNA methylation by immunofluorescence, they couldn't present any discrepancies between frozen eggs and fresh eggs. And another technology, looking for an expression in humans of on impact of vitrification by microarray, again, they couldn't present any discrepancies. So at least we are relieved, because otherwise it's catastrophe. If every cryopreservation process is going to do these changes, we are in big trouble. This is part of our everyday life in our reproductive medicine and the treatment of ART. This literature review highlights that there is a need to learn more about regulatory mechanisms. So the current investigation by new technologies to perform the large-scale analysis of DNA methylation by methylome, we discussed it, and transcriptome, from a small amount of material as little as single cell. This is going to give the answer to this heavy question. Fatigue to 2020, reproductive technologies, female infertility, and the risk of imprinting disorders. This is a very big study that was done in Paris, in France. They took 3.5 million pregnancies, singletons, that were born after natural conception, assisted reproduction, ovulation induction, thawed, frozen, fresh, and so on. And actually, they were looking on risk of imprinting-related disease according to mode of conception. And when they looked into the fresh ET, they found that relatively to natural conception, the odds ratio was significantly higher. And in endometriosis, again, much higher. This is very interesting. This is a new aspect. We didn't know that endometriosis per se is causing these changes, epigenetic changes. And when they looked into imprinting-related disease in natural conception versus those with endometriosis, it was much higher. And fresh embryo versus natural conception, again, exactly the same. And another thing, They looked the application of endometriosis or non-endometriosis on the newborn. And they checked the newborn diabetes mellitus. We spoke about it yesterday as well. And they presented that in natural conception, in the newborn diabetes mellitus was higher with endometriosis and as well in fresh embryo transfer. So the fresh embryo transfer, 1.58, endometriosis, 1.47. This prospective epidemic, it's tremendous study because it's a very huge population, 3.5 million. This prospective epidemiological study showed that the risk of clinically diagnosed imprinting-related disease is increased in children conceived after fresh embryo transfer or from mothers with endometriosis. This is absolutely a new aspect. Again, our dear Boris Novakovic, I will not repeat his data, but he showed these fantastic results that neonates that were born with these markers of epigenetic markers, where there were adults actually, he couldn't find anyone. Again, the question is what's happening after another 10 years, another 10 years, and so on. Is good. Psychological distress. Again, we're coming back to psychology. Psychology, psychiatry, it's tremendously important. The impact of psychological distress during pregnancy on the developing fetus, biological mechanisms, and the potential benefits of meaningful interventions. And they have a scheme how it happens. Perception of stress, paraventricular nuclei in our brain that are producing corticotropin-releasing CRF, and CRF goes in two arms. One arm is sympathetic adrenomedullary, and the other arm, which is important for us, is hypothalamic pituitary adrenal. And by releasing ACTH, they release cortisol. Their hypothesis is when we have a chronic or even acute severe stress of the mother during the pregnancy, the glucocorticosteroid receptor expression is downregulated, downstream stress response. This causes in the mother secretion of the cortisol. The cortisol is crossing the placenta, and as I mentioned before, the NRC31 methylation actually is produced. GR expression is reduced, and negative effect on growth and development. So the in vitro, the in utero environmental plays an essential role in shaping future growth and development. Richmond is looking at the folic acid. We discussed yesterday folic acid with our Italian colleague. And this is a fantastic study that was done in Aberdeen many years ago. They took women, pregnant women, that part of them got 0.2 milligrams of folic acid, and part of them 5 milligrams of folic acid. I want to tell you that I give to my reproductive patients, all of them are getting 5 milligrams of folic acid. I really think that overdose is better than too lower dose. And because we have more congenital malformation in children after ART, I want to be sure that this part is covered. The better indicate the long-term impact of maternal folic acid in pregnancy on DNA methylation of offsprings. And it was done by Epigenome-White Association study. And they checked the children after 47 years by saliva samples, Illumina Infinium human methylation, 450 area. And they presented there are tremendous changes in 46 regions in terms of methylation and demethylation in children that got 0.2 versus 5, with much less changes in 5 milligrams. So, these results of the study suggest that maternal folic acid supplement uses associated with changes in the DNA methylation of the offspring that persist for many years after exposure in utero. Black and white. Ciofani, 2019. Impact of assisted reproduction, infertility, sex, and paternal factors on placental DNA methylome. Professor Glazerman spoke about it yesterday, another about the placenta. And they checked for epigenetic changes in control placentas, 44 placentas that were for natural conception, and after ART. And they found that when they looked into DNA methylation levels, all the markers in vitro, in placenta, they were much, much higher. Placenta is susceptible to the induction of epigenetic abnormalities by ART. Within the ART group, subgroups could be identified by DNA methylation clusters analysis, differentiated according to infertility and type of ART procedure, and affecting numerous genomic regions, including those involved in cellular and nervous system development and function. So maybe it's going to be classified by subgroups, depends on what sort of infertility treatment was done. So the take-home messages. Epigenetic is a science of heritable phenotypic changes that do not involve DNA. Such effects on cellular and physiological performance may result from external or environmental factors or to be a part of normal development. Mechanisms that produce such changes are DNA methylation and histone modification. Gene expression acts by repressor proteins that attach to silencer regions of DNA. The impact of epigenetic imprinting can be expressed in twins pregnancies, in diseases, cancer, diabetes, and conditions such as stress, addiction, and depression. And the role of HCG, anticoagulants, progesterone, and IVIG is still unknown. Epigenetic changes can be sporadic or transgenerational. Fetal programming has a tremendous impact on developmental origins of health and disease in the adult, and lifestyle has a great impact of epigenetic changes and late pregnancies. And finally, some epigenetic markers show that the neonatal period resolves spontaneously in ART and non-ART born adults, and psychological distress during pregnancy can cause cognitive and behavioral problems during childhood and adult life. And I always finish my lecture with the slide of my grandchildren. So if we have a second, I will tell you, this is our biggest grandchild, Noya. Noya is 16 years old, and her name is composed of two parts. Noi is beauty, and the I is God. She is our beauty of God. Yair is 14 years old, and his name is the beam of light. And this is the little Neta. And these children are beam of light in our lives. Thank you very much.